just a quick summary of what Bjorn showed us. So with aging, the brain undergoes enhanced sclerosis, and then we experience stiffening of the tissue. OPC can sense this mechanical alteration, which leads to impaired regenerative capacity. And the question I'd like to ask is whether the nuclear lamina remodeling due to OPC mechanotransduction may alter laminar chromatin interactions that may feed back to impaired regenerative capacity if we can target that. So I'll focus on microrheology of the cell nuclei. We have an extracellular mechanical input, and this extracellular mechanical input is sensed by the cells. There is a remarkable phenotype or behavior of mammalian cells that the intracellular mechanics, which I refer to as the nucleus mechanics, is coupled to extracellular mechanics. And this is true with stem cells that have soft nuclei that undergo stiffening as, for example, embryos age and extracellular mechanics increases in a mechanical, sensible fashion. And in order to probe that, so what you see here is H2B cherry, and we apply micropipette aspiration. It's a very neat approach. What I like about it is how simple it is. So basically, we use a pipette, we apply suction, and this is a constant pressure that we apply on the cell and or on the nucleus. I'm not going to go into details. And then we look at the stress-strain relationships, and we can obtain viscoelastic properties from this experiment. We recently showed what we term a unified model of the nucleus, of the nucleus mechanics. But basically what we find is that nuclei respond as follows. When you apply suction, or when you apply mechanical load, they will undergo deformation, which is instantaneous. This is marked by this spring here. And then there will be a viscoelastic response, which is modeled by this Kelvin-Voigt module. And then there will be a consistent flow, a fluid-like behavior response to stress of the nuclei. And this is true for a wide range of nuclei and cell types. When we look at a cell, this is how it looks like. And this is the linear viscoelastic model we use. So an experiment basically looks something like that. I don't know if you can see that. So what you see is an OPC. It looks distorted just because in the medium we add cytocalycin D in order to depolymerize the actin cytoskeleton. And we try to detach the cell, although these cells really grab strong using trypsin. So what you should have seen here is that when suction is applied, we pull on the cell and mostly on the nucleus within this OPC. And the creep compliance, the response looks like that. It adheres to the model I showed you before. And when you analyze that, you find that in terms of nucleus mechanics, it's not much different than fibroblasts. The steady state mechanics or steady state stiffness is comparable to embryonic stem cells. But what we do find is a major increase in viscous terms. So viscosity is exceptional. In order to test that, what we use is a different approach, a nano indentation rheology. It looks something like that. What you see here in the middle is a nucleus with a probe that's coming from the top and pushes on that, again, to study the stress-strain relationships of these cells. And what we find is this kind of response, which can be modeled like that. And again, what we find is mechanical response, which is similar to other cell types, but a major increase in viscosity. Now, from our experience, increase in viscosity of the nucleus is associated with decondensation of chromatin. And this is our next task. We are going to study how extracellular mechanics aging related to stiffness affect nucleus viscosity. Specifically, If viscosity marks indeed increased decondensation of the chromatin, we would like to map that, to profile that using various approaches like DEM-ID or cut and run technologies that we develop in lab. And then we would like to see whether nucleus mechanics could be a therapeutic target in order to prevent these aging effects. And I will conclude here. 2 minutes for questions. Thank you very much. Thank you.
Okay, so I would like to thank Bjorn Neumann and the Amnon Buxboim for the talk.